My name is Thomas Crabtree and I'm a Diabetes and Endocrinology Registrar and Clinical Research Fellow working at the Royal Derby Hospital in the East Midlands and Sandwell and West Birmingham Hospitals in Birmingham, and I'm grateful today for this opportunity to present this analysis from the ABCD SGLT2 inhibitor audit programmes assessing the effect of these drugs across the class on urinary albumin concentrations. These are my disclosures. The ABCD SGLT2 inhibitor audit programs first began in 2014 with the dapagliflozin audit program. Subsequent to this, in 2016 and 2017 respectively, the canagliflozin and empagliflozin audits were launched. The aims of these audits, as with all audits, was to collect anonymised routine clinical data for patients taking SGLT2 inhibitors in order to gain real-world data on their use. Data for these audits is received from primary and secondary care via the online audit tool, or from primary care via data submitted by clinical commissioning groups in bulk. We know from randomised control trials that SGLT2 inhibitors are efficacious in preventing hard renal endpoints. Less attention has perhaps been paid on the impact that these drugs can have in reducing urinary albumin levels, which is obviously an intermediate effect that they will have in preventing these hard renal endpoints. Bay et al. Performed a systematic review and meta-analysis utilising data from many of the randomised control trials that we have grown accustomed to, looking at sub-analyses focused specifically on the impact on urinary albumin levels and demonstrate a cross-class effect in reducing the urine albumin creatinine ratios. This analysis in particular included the drugs that we're going to be looking at today: canagliflozin, dapagliflozin and empagliflozin. The forest plot on screen at the moment demonstrates the results of that meta-analysis. Although we obviously give a lot of attention to randomised control trial data and data in the form of meta-analysis from randomised control trials, that's one part of a bigger puzzle, and there's still a place for real-world evidence, which can be beneficial because it provides insights into benefits in situations where patients are perhaps less intensively followed up, where there is perhaps less additional support available to them, or where they may have more extreme baseline characteristics than those patients allowed to be enrolled in clinical trials. Previous real-world analysis from the ABCD audits looking specifically at empagliflozin demonstrated significant reductions in albuminuria across the population, with the caveat that this analysis focused on absolute rather than relative reductions. In those with a urine albumin creatinine ratio greater than 300, the reductions were very large, with an average reduction of 379.3 micrograms per milligram. To our knowledge, there's limited evidence available at present comparing the efficacy of different SGLT2 inhibitors in the real world on the outcomes related to urinary albumin creatinine concentrations. With this in mind, the aim of this analysis was to assess changes in urinary albumin creatinine ratios across the SGLT2 inhibitor class and to look for any particular difference between the drugs. For this analysis, data were extracted from the ABCD SGLT2 audit programs, and the analysis was conducted in Stata 16. To be included, patient datasets required at least a baseline dataset for urine albumin creatinine ratio and one follow-up. We looked at both absolute changes in urine albumin creatinine ratio, but we also looked at relative changes expressed as a percentage reduction from baseline. We did a stratified analysis where we grouped patients by normo, micro or macroalbuminuria at baseline, and we also grouped them by SGLT2 inhibitor drug to allow for comparisons between the three drugs that we had data for. There's significant skew to urine albumin creatinine ratio data, and therefore we utilised non-parametric methods. We used the Wilkinson-Stein rank test to assess for within-group changes, and we used the Dunn's test with Bonferroni corrections to look for differences between the different groups. These are the baseline characteristics of our cohort on screen at the moment. Our cohort was followed up for a median duration of 1.7 years with an interquartile range of follow-up between 1 and 2.8 years. For reference, I've put the baseline HbA1c and BMI of those included in the EMPA-REG renal sub-analysis just to demonstrate that there is a difference between our cohort and that cohort, and As is often the case with real-world data, our cohort had much more extreme characteristics as baseline, which we'll look at in a second. You can see that between each SGLT2 inhibitor drug, they had broadly similar ages, broadly similar baseline BMIs, and broadly similar HbA1cs, with the BMI and HbA1cs being more extreme than those observed in the randomised controlled trials. You can also see that there was comparable urine albumin creatinine ratios across the groups, as well as comparable eGFRs at baseline. The results of the analysis are on screen now. Across the population as a whole, looking at change in urine ACR in absolute terms, reductions were observed across the population with a p-value of 0.018. There was a small increase in urine albumin creatinine ratio in normoalbuminuric individuals at baseline, with an increase of 0.1 micrograms per millimole, and this was statistically significant, although the clinical relevance of such a small increase is uncertain. In those with microalbuminuria and macroalbuminuria at baseline, there were significant reductions in absolute terms in urine albumin creatinine ratio between baseline and follow-up, and these are demonstrated in the bar chart on screen now. In relative terms, this remained similar, and the largest reductions, again in relative terms as well as absolute terms, were in the macroalbuminuria group, with a 55% reduction in urinary albumin levels between baseline and follow-up. We then went on to compare the different drugs. I haven't included normoalbuminuria here because the change was small and in the opposite direction, but we've looked at both microalbuminuria and macroalbuminuric individuals at baseline and compared the change in urine albumin creatinine ratio with each drug, and the bar charts very much speak for themselves, and you can see that with dapagliflozin there was attenuated reductions in urine albumin creatinine ratio, but there was no significant difference between canagliflozin and dapagliflozin in terms of the impact of these drugs on urine albumin creatinine ratios in either of these two groups. So, to conclude, this analysis demonstrated reductions in albuminuria across the class. The largest reductions in urine albumin creatinine ratio in both absolute and relative terms were observed in those people with macroalbuminuria at baseline. Dapagliflozin was associated with slightly smaller urinary albumin creatinine ratio reductions in people with microalbuminuria and macroalbuminuria at baseline, and the reasons for this aren't clear. Obviously, we have the caveat that this is observational data, so there may have been potential confounders which we haven't measured or identified that we're unable to correct for in this analysis. Additionally, in this analysis and in our work, we had much smaller numbers of users using dapagliflozin, so it may be that we've just not managed quite to capture the extent of the change with dapagliflozin with a slightly smaller data set. Thank you for taking the time to listen to this presentation. If you'd like to learn more about the ABCD audits, you can follow the link on screen now. We have several audits ongoing at the moment, including audits looking at semaglutide in both injectable and oral forms. The SGLT2 audits are of course ongoing as well, and we have audits looking at diabetes technologies, which include Freestyle Libre, DIY artificial pancreas systems, closed loop systems, and this week at Diabetes UK we'll be launching the ABCD Omnipod audit, which we would welcome you registering for. Again, thank you for taking the time to listen to this presentation, and I welcome any questions that you may have. Thank you.